Hello, and thanks for joining me at this odd hour. A major theme of the synthesis era, going back to Fisher, was the rejection of non-Darwinian theories, including internalist theories that refer to internal tendencies of variation. For instance, when Simpson reconciled paleontology with this emerging orthodoxy, he looked at apparent trends in the fossil record. He argued that they probably weren't real trends. And then he concluded that even if they are real trends, they must be due to selection because the only alternative is vital urges or cosmic goals without known mechanism. Now, where did Simpson get this confidence when generations of thinkers had simply assumed that if organisms tend to vary in a particular way, that this would cause an evolutionary tendency? We know the answer to this question. Simpson and the other architects of the modern synthesis believed that this kind of causation was ruled out by the harsh mathematical logic of population genetics, because in fact, Haldane and Fisher had said that because mutation rates are small, mutation is a weak pressure easily overcome by the opposing pressure of selection. It will only be important if mutation rates are abnormally large or if selection is absent, if we're talking about neutral characters. Haldane argued this in 1927. Fisher followed up in 1930 with a stronger version of the argument, and it was repeatedly cited by leading thinkers. So here is Gould, 75 years later, saying, since orthogenesis can only operate when mutation pressure becomes high enough to act as an agent of evolutionary change, empirical data on low mutation rates sound the death knell of internalism. Because this argument, of the way this argument eliminated an entire class of internalist theories, Provine identifies it as a key claim in his 1978 paper on how theoreticians contributed to the synthesis. Because the Haldane-Fisher argument is so important, it's also important to understand that the argument is wrong. An evolutionary process that depends on events of mutation that introduce new alleles will be subject to biases in the introduction process by a simple first-come, first-served logic that Haldane and Fisher did not consider. This kind of causation, linking tendencies of variation with tendencies of evolution by way of biases in the introduction process, was first proposed and explained formally in 2001 and demonstrated using computer simulations of a simple population genetic model along with some mathematical arguments. Subsequent theoretical work, which Joanna Mosel is going to review, has shown that this kind of causal link emerges under broad conditions. It's not a special case. Empirical studies that David McCandlish will discuss have confirmed a key prediction, which is the effect of simple mutation biases such as transition-transversion biases on the changes involved in adaptation. Contrary to Haldane's conclusion, this effect does not require high mutation rates or neutral evolution. This kind of causal link makes it possible to specify a rigorous causal grounding for key historical themes of internalist thinking. Attacks on specific evolutionary propensities, trends due to internal tendencies, intrinsically likely forms, and to rebut objections to these themes like the Haldane-Fisher argument, and to pose new and specific testable kinds of hypotheses. So that's all very exciting scientifically. But for my talk today, I'm going to focus on a meta-scientific issue of why it took so long for this kind of causation to be recognized. It was literally absent almost completely from 20th century thinking. Why is that? What does that tell us about the process of science, how scientists think, how they don't think? And rather than answering this question, I am simply going to attempt to establish the premise by pointing to 4 places in evolutionary discourse where the absence of this theory, retrospectively, is conspicuous. And in the process of doing that, I'll explain some of the broader implications. So the first case is simply the classic rejection of internalist theories on the basis of the Haldane-Fisher argument. So as I said, Haldane concludes that mutation can be important only for neutral evolution. That's what he means when he says factors of negligible advantage or disadvantage, or when mutation rates are abnormally high. And Fisher was more dramatic, characteristically. For mutations to dominate the trend of evolution, it is thus necessary to postulate mutation rates immensely greater than those which are known to occur, and he concludes that all of these internalist theories have to be thrown out once you accept Mendelian inheritance as the basis of evolution. So this argument was frequently cited. I won't read these because it takes too long. Haldane, Fisher, Wright, Huxley, Ford, Simpson, Meyer were some of the leading thinkers who employed this Haldane-Fisher argument. And anytime we see this argument without a rebuttal saying that it's wrong, that indicates that the theory of biases in the introduction process is missing, is absent. The second case refers to hypotheses of mutational effect in molecular evolution. So this plot comes from the original 1969 co-proposal of the neutral theory by King and Jukes, and it shows a very strong correlation between the frequencies of amino acids in protein sequences and the expected frequencies from applying the genetic code to random sequences, randomly generated sequences. So most of the effect comes from the number of codons assigned in the genetic code. So methionine and tryptophan, down here in the lower left, have only one codon each. Serine, leucine, and arginine, which is a bit of an outlier, have 6, and then there's the ones with 2 here, isoleucine has 3, and all of these up here have 4. So this is the kind of expectation one would get if evolution is just mutation without any selective filtering. And on this basis, King and Jukes presented this correlation as evidence of neutral evolution. And more generally in the molecular evolution literature, mutational explanations for patterns are typically assumed to imply or to require neutrality. I could give a lot of examples. I'm going to just read one here where Lafay and colleagues, talking about strand-specific skew effects, say, these changes in amino acid and codon compositions represent neutral sequence change that has been caused by strong strand and species-specific mutation pressures. So this assumption, when you see this assumption that mutational causes for patterns require or imply neutrality, that indicates that the theory of biases in the introduction process is missing, because the effect under that theory does not require neutral evolution. Okay, but there's a really interesting twist to this story that I want to tell you about now. So Jack King was one of the co-proposers of the neutral theory and the author of that 1969 paper, and he almost immediately recanted his position and said that this codon correlation that I showed you was not evidence of neutrality. And I'm just going to read out his argument here. He says, if a gene is in the process of progressive adaptive evolution, there might very likely be more than one among the 1,000 or so possible single-step changes that would be evolutionary advantageous. Then the first of these to occur by mutation would have the first chance to take over. So you see, he sees that first come, first served logic. He gets it. The conditions of selection would then be changed, and it would be too late for the other previously potential candidates. Thus, the probability of fixation, and here means probability of evolution, is going to be a function of frequency of arising by mutation. And over time, the distribution of amino acid frequencies will reflect, more or less passively, the peculiarities of an arbitrary genetic code. So he gets the thing that this is not about facilitated variation, even an arbitrary code, even if most evolutionary changes are due to exclusively Darwinian adaptive evolution. So clearly King, in 1971, when this was published, has an intuitive sense of how the possibility space for evolution shapes outcomes by inducing a kinetic bias, a first come, first served effect, in what is proposed by mutation. And he explains this with a clearly worked out example. However, King doesn't generalize this into either a population genetic theory of this first come, first served effect, or a theory about how genetic spaces induce biases in mutation. And so this argument seems to have been a dead end. This particular paper was cited just 7 times. He has another paper from the same year or a year later where he makes some similar arguments, and that's been cited a couple dozen times. Before continuing, I want to use this example to explain some basic principles about developmental biases using the genetic code, because the genetic code is a genotype-phenotype map. It relates codon genotypes to amino acid phenotypes. And if we start with this methionine phenotype, there's 4 different amino acids shown here that can be reached by a single nucleotide mutation. Leucine can be reached by 2 different single nucleotide mutations, and isoleucine can be reached by 3 different single nucleotide mutations. Meanwhile, these amino acids shown in gray here, like tyrosine and cysteine, they cannot be reached by any single nucleotide mutations. It would take a double or a triple change. So in this way, the genotype-phenotype map induces 4 different degrees of mutational accessibility of alternative phenotypes, starting from the focal phenotype of methionine. So even if all mutation rates are the same, so there's no mutation bias, there's still going to be this phenotypic bias or developmental bias that's induced by the genotype-phenotype map. This is the local effect of accessibility from a focal genotype. But there's also a global or long-term effect due to the fact that some phenotypes have more genotypes. So for instance, leucine has 6 codons, whereas methionine only has 1 codon. So leucine has more mutational arrows pointed at it from other codons, from other parts of sequence space. That makes it more findable by an evolutionary process that searches the possibility space by mutation. So Jack King's revised codon argument is a case of this global effect of findability due to the number of genotypes assigned to a phenotype. And more generally, any non-trivial genotype-phenotype map is going to induce these same two kinds of effects. There's a local effect. So this is kind of a stylized map of RNA folds mapped to sequence space. So there's 3 different folds here, and starting with the gray one, make that the focal phenotype. The green fold is more accessible because there's more neighboring genotypes than the blue one. But the global effect of findability favors the gray phenotype because it has about twice the surface area, twice the contour length as the other ones. Okay, so moving on to the third case. This involves the literature of developmental constraints. So references to developmental constraints typically lack an explanation of how developmental effects on variation are supposed to influence evolution. So this gap in the implied chain of causation goes back to the seminal 1985 paper by Maynard Smith and colleagues, which has been cited now almost 1,700 times. In fact, interestingly, Maynard Smith and colleagues recognized this gap. And they drew attention to it. They're asking here, the issue is whether biases on the production of variant phenotypes, which they call developmental constraints, such as those just illustrated, cause evolutionary trends or patterns. And then they cite the Haldane-Fisher argument as an impediment to proposing that. And they say this position deserves re-examination. Now, they don't actually offer a re-examination. They just suggest neutral evolution, which is not a satisfactory grounding for evo-devo. So critics then attacked that. So in their defense of the adaptationist research program a few years later, Reeve and Sherman just responded by criticizing. They criticized Gould and Lewontin, and they criticized Maynard Smith et al. By saying there's no delineation of a mechanism. They're just keep restating this idea of developmental constraint or developmental bias. And they went on to ask, well, why wouldn't selection suppress an easily generated trait? So they're basically calling on the Haldane-Fisher opposing pressures argument to rebut the developmental constraints argument. And this was not the only argument used against developmental constraints. There were a lot of versions of this causal status objection that Meyer is using here, where he says, well, evolution is about evolutionary causes, but development is about its approximate cause. It's not an evolutionary cause. And there are a number of examples of this in the literature. So in this way, the literature is providing us with a debate between scientists. This is a documented debate between scientists over the causal basis of developmental constraints. And the disagreement that they're having here could have been resolved by combining the efficacy of biases in the introduction process with the argument that I just gave you about how a genotype-phenotype map induces differential mutational accessibility of phenotypes. Okay, final example. A classic internalist theme is that evolution makes use of forms or structures that are intrinsically likely. And Kaufman made a version of this argument in regard to things like regulatory networks. He argued that certain properties emerged in evolution simply because they're more common in abstract possibility spaces. His name for this effect was self-organization. He said again and again that selection and self-organization were partners in evolution and worked together in the origins of order. But because he didn't explain this partnership in terms of classic forces, in terms of standard evolutionary forces, population geneticists did not know what to make of his arguments. For example, one reviewer quoted here depicted his references to self-organization as being magical. And there have been many subsequent arguments of this type. The most compelling, there's a very empirically compelling argument about RNA folds that's shown here. So this plot from a recent study by Dingle and colleagues shows the frequency of RNA fold classes from folding up lots and lots of random sequences. And so the frequencies for the top 45 ranks here are shown. The ones that are circled in yellow are the ones that are found in nature. So RNA folds found among natural RNAs, naturally evolved RNAs, are extremely likely to be the most common folds in sequence space. However, the literature on findability presents this effect as the consequence of a background condition or as a property of the fitness landscape. That is, just like the case for developmental constraints, there's a gap in the implied chain of causation here, in which findability is attributed to, again, a background condition rather than to some change-making causal process. But I already gave a causal explanation for this. This case is precisely analogous to King's codon argument. So whenever we see findability arguments, without a complete causal explanation that refers to population genetics, this suggests that the theory of biases in the introduction process is missing. I'm going to skip over this interesting passage where John Stuart Mill sort of invokes biases in the introduction of truth to explain something. It's an interesting passage to look at. So why did a theory of biases in the introduction process as a cause of orientation or direction take so long to emerge? Literally, 70 or 80 years after the founding of theoretical population genetics. I will leave that question to you, historians and philosophers. I hope that I have piqued your interest in this question by establishing the premise that this theory is indeed absent from the evolutionary discourse of the past 80 years. Thank you, and I welcome your questions.